Okay, the next point is a dihybrid cross. So what is dihybrid cross? A cross between two pure homozygous parents by considering two pairs of contrasting character at one time. In a monohybrid cross, we consider only one pair of contrasting character, and in a dihybrid cross, Mendel considered two pairs of contrasting characters at one time. For example, Mendel crossed a pea plant having characters like rounded seeds and with a yellow color. So these are the two characters. Seeds, these are the round and the yellow in color. This plant crossed with a pea plant having green seeds and wrinkled seeds and the color is green. So the wrinkled seeds with a green color. Here round is dominant over the wrinkled and yellow color of seed is dominant over the green. Okay. So these two plants, they are homozygous or pure in a character. So these plants, they cross. See, these are the parents, parental generation. Genotypes of these parents, these are the capital R for the round and the capital Y for the A. In the same way, for the genotype for the wrinkled and green, small r for wrinkled and small y for the green. Then this one of the parent considered as a male or one of the parent is considered as a female. So this individual, they produce gametes. The gametes of the genotype is a homozygous individual. So therefore capital R is homozygous. They form the gametes, that is capital R and Y. So these gametes are formed, that is R and Y. Capital R, capital Y. Here gametes are formed, it is small r, small y. Okay. Then these gametes, male and female gametes, they fuse together to form the F1 generation. In F1 generation, individuals are obtained, this individual called as F1 hybrid, or it is also known as a dihybrid. Mendel observed that in a dihybrid or in a F1 generation, all plants, they having a round shape or rounded seed with a yellow color. So the round and yellow seed, these characters or these traits that appear in F1 generation. So these recessive traits are not appear. So the law of dominancy follow this dihybrid cross also. So dominant characters, that is round and yellow, is appear in F1 generation. Then he selfing the F1 hybrid, they self to form the gametes. How to make gametes here? The capital R, small r, capital Y, small y. See here, I give one trick, friction method. This is known as capital R and this is the small r. So this capital R cross with the capital Y, then cross with the small y. So capital R, capital Y. This is one type of gamete. Then this is this capital R and this small y. This is the another type of gamete. In this case, small r, capital Y, small y. So small r, capital Y, small r, small y. So four different types of gametes we get. So this method we use for the formation or for making different types of gametes. Okay. Then these gametes are formed. These gametes, by using checkerboard, the F2 generations are obtained. In an uppercase, you write the male gametes. In lowercase, on left hand side, you write the female gametes. So these gametes, they fuse together to form the different individual. There are 4 gametes are formed. So this genotype, capital R, small r, capital Y, small y, this genotype or the individual having such type of genotype, they produces 4 different types of gametes. So these gametes are to keep in a Punnett square checkerboard and to obtain the F2 generation. See here, in a first box we write capital R, then small y, small y. This individual, it is round A. This is the round A, it is homozygous round. So this one is a homozygous or a pure round individual. Then capital R, capital Y, small y. So capital R and capital Y, therefore it is also round A. It is also round L. Capital R, small r, capital Y, it is also round yellow individual. So rounded seed and yellow color, the dominant trait is occur. Then here also capital R, small y, therefore it is also again a round yellow. Then this case, this offspring, it having capital R, capital Y, small y, so it is also round yellow individual. See here again, capital R, small y, both are dominant, therefore it is also round and yellow. This also round and yellow individual. This individual in which one again is capital, another is recessive, again Y is capital, another is recessive. So this is again round yellow. Okay. Then this is round yellow. Okay. So 1, 2, 3, 4, then 5, 6, 7, 8 and 9. 9 individuals we get. These are the having seeds. These are the round and yellow color. The seed having round shape and yellow color. So these 9 individuals we write here. Phenotypic ratio of this, that is a 9. Okay. Then here small r, so capital R, small y. So this is individual is having seed, rounded seed with a green color. So this new combination is of rounded seed with a green color. Small y we denoted for the green. It is a recessive trait. So the round and green trait is occur in this cross, in this box. Here also small y for green, capital R, small y for the round. So this is again round green individual. So this is round green. This individual is again round green individual. So the 3 individuals, these are the round green individuals. Here we count this, then this 3 is completed. Then this one, small r and capital Y, small y. So small r for the, we denote for the wrinkled. So wrinkled and capital Y, small y for the yellow. So wrinkled yellow individual. This one. Then this individual, small r for wrinkled, capital Y for the yellow. Wrinkled yellow. This again wrinkled yellow. And these are the homozygous recessive. These are the wrinkled green. Okay, these are the wrinkled green individual. Only one wrinkled green individual is formed. So, if you observe this Punnett square, a few parental traits are occur. The parental traits, only two parental traits, that is homozygous round yellow and homozygous wrinkled green. These parental traits are occur, and new combinations or new recombinations of the new traits are occur. These are the round green and wrinkled yellow. Three new traits or new combinations are occur in a dihybrid cross. So phenotypic ratio, you write this phenotypic ratio. 9 individual, they having rounded seed with a yellow color. 3 individuals, they having rounded seed with a green color. 3 individual, they having wrinkled seed with yellow color, and only one with this individual having a wrinkled seed with a green color. So the phenotypic ratio of a dihybrid cross is a 9:2, 3:2, 3:1. Remember this phenotypic ratio. And the genotypic ratio is 1:2, 2:2, 2:4. As to 1, as to 2, as to 1, as to 2, as to 1. So this 1 means the capital R, capital Y, capital Y. Then 2, this capital R, capital Y, small y. Similar genotype we find this capital R, capital Y, small y. So these two individuals, in this way, we have to find the genotypic ratio also. So the genotypic and phenotypic ratio, they are different. Phenotypic ratio is a 9:2, 3:2, 3:1, and the genotypic ratio is 1:2, 2:2, 2:4, as to 1:2, 2:2, 2:1. So totally 16 individuals are formed in a dihybrid cross. Okay. Here, by using dihybrid cross, Mendel gives a third law. The third law of inheritancy is known as a law of independent assortment. So third law is a law of independent assortment. Okay. By using monohybrid cross, we learn two laws. First is the law of dominancy, and second is a law of segregation. And by using dihybrid cross, Mendel gives law of independent assortment. So what is the law of independent assortment? According to the Mendel, when dihybrids in F1 generation, they form gametes. These dihybrids formed in F1 generation, they get selfed, and during formation of gametes, these alleles, this capital R small r, capital Y small y, these alleles, they get segregated, or the factors, they get segregated or separate from each other, and they segregate from each other and they enter into the different gametes. So these factors or the alleles in a dihybrid, they segregate or separate and assort independent with each other. So the assortment means the distribution of gamete is independent of each other. So this capital R, it goes with the small y, capital R distribute with the y. So gametes are different. Gametes are formed capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So different gametes are formed. So the factors or the alleles, they segregate and assort independent with each other, and therefore in the F2 generation different new combinations are occur, and we get the ratio 9:2, 3:2, 3:1, in which 9 individuals they shows these are the round, they show the rounded seed, rounded seed with yellow color. The three individual, they show the rounded seeds with the green color. Three individuals are, these new three combinations are formed. These are the three rounded seed with the green color, ring colored seed with the yellow color. So these two combinations, new combinations are formed due to the assortment of these factor of the allele. So third law of dominancy was explained by using this dihybrid cross. This dihybrid cross is only applicable when the factors of these genes, they are present on separate chromosome. If these alleles or these factors, they present on the same chromosome, then they become linked up with each other, they show the linkage, and then they do not inherit together. Therefore, remember this one thing is very clear. The law of independent assortment is applicable only when the factors or the alleles, they are located on a separate chromosome. Okay. So that is about the dihybrid cross and the law of independent assortment. Then here for some exam, NEET or for CET, some questions are asked. What is phenotypic ratio of dihybrid cross, or how many individuals are formed in a dihybrid cross? They are having genotype like capital R, small r, capital Y, small r. So question asks in exam like this: how many individuals are formed in dihybrid cross? They are having genotype as a capital R, small r, capital Y, small r. Then how to remember this thing? See here, capital R, small r, capital Y, small r. This individual, how many individuals they are formed? So I give some trick here. Here you write 2 raised to n. 2 raised to n means 2 and n is heterozygous. N for the heterozygous condition. Here capital R, small r is heterozygous. Capital Y, small y is also heterozygous condition. So 2 into 2. 2 heterozygous condition, therefore you write 2 into 2. So the answer is 4. The 4 individuals are formed in a dihybrid cross. They are having genotype capital R, small r, capital Y, small y. In the same way, for example, here also another trick. Small r, small y, small y. How many individuals are formed? So all are homozygous individual, they are formed only one time. So small r, small y, small y. These individuals are formed in only one time in that out of 16 individuals. Capital R, capital Y, it is also homozygous. They also found in only one time. Then this capital R, small r, capital Y, small r. How many times these individuals are formed? So only one is the heterozygous condition. So 2 raised to n, 2 raised to n is 1. So only 2 individuals, 2 into 1, that is 2. So the 2 individuals are formed. They are having genotype like capital R, small r, capital Y, capital Y. See here, 2 individuals, find capital R, small r, capital Y, capital Y. This one individual and another small y. Here also another individual. So these 2 individuals, only 2 individuals are formed. They are having genotype like this. So use this trick to solving this problem. I think you understand the dihybrid cross, the phenotypic ratio is important one, and law of independent assortment.